Good morning, students. Welcome to the e-learning platform by Department of Collegiate Education. In today's video lesson, let us learn about germ layers. In our previous class, we learned about the body organization, which is seen at different levels, that is at organ, cell, or subcellular level or acellular level, protoplasmic level, followed by the cellular level, tissue level, and organ system level of organization. The protoplasmic grade of organization is seen in phylum Protozoa. The cellular grade of organization is seen in poriferans like sponges. Tissue grade of organization is seen in coelentrates like Hydra, in sea anemones and other jellyfishes. The organ grade of organization is seen in phylum Platyhelminthes, and the organ system grade of organization is seen in phylum Annelida, Arthropoda, and other higher phyla. So this is what we learned in previous class. And in today's chapter, we'll try to understand the concept of germ layers, the different types of germ layers, and the importance of germ layers. And by the end of the session, you'll be able to understand the importance of germ layers and the organs derived out of germ layers. So what is germ layer? Germ layer is a primary layer of cell that formed during embryogenesis. So what is embryogenesis? During the formation of or the development of zygote, it undergoes various stages. So the embryo is being formed. That is what is known as embryogenesis. That is, zygote undergoes division to form a morula. Morula further divides to form a blastula. Blastula further divides to form a gastrula, and so on. So this is the process of embryogenesis. During this embryogenesis, a primary layer of cells are formed. That is what is known as germ layers. The three germ layers in vertebrates are particularly pronounced. However, all eumetazoans, that is animals more complex than sponges, they produce 2 or 3 primary germ layers. Some animals like cnidarians, they produce 2 germ layers, that is the ectoderm and the endoderm, making them diploblastic, di, that means 2 layers, that is ectoderm and endoderm. Whereas other animals like chordates, they also produce a third layer of cells that is known as mesoderm. Between these 2 layers, between the ectoderm and endoderm, you have the middle mesoderm. So therefore, it makes them triploblastic. Germ layers eventually they give rise to all of animal tissues and organs by the process of organogenesis. Here, I've taken the gastrula of both diploblastic as well as triploblastic animals. In diploblastic animals, you have the ectoderm and the endoderm, whereas in triploblastic animals, another extra mesoderm layer is present between the ectoderm and the endoderm. And the process of formation of germ layers is seen in the gastrula stage. So during the process of gastrulation, the germ layers are formed. So therefore, I've taken example of or the image of gastrula. Among animals, sponges show the simplest organization, having a single germ layer. So sponges have only one germ layer and they have the simplest organization. Although they have differentiated cells, that is, cells are getting differentiated into collar cells, cornoblast, pinacocytes, etc., they do not have any tissue coordination. They absolutely lack tissue coordination. Whereas diploblastic animals like cnidarians and ctenophores, they show an increase in complexity, that is, they have 2 germ layers, the ectoderm and the endoderm. The diploblastic animals are organized into recognizable tissue. So you find tissue grade of organization in these diploblastic animals. And all higher animals, from flatworms to human beings, they are triploblastic in nature. That is, they have an extra cell layer called as the mesoderm, in addition to the germ layers, ectoderm and endoderm. So in between the ectoderm and endoderm, you find the mesoderm. Triploblastic animals develop recognizable organs, which is lacking in diploblasts. So based on the number of germ layers, we have 2 conditions, that is diploblastic condition and triploblastic condition. Diploblasty is a condition of blastula in which there are 2 primary germ layers, the ectoderm and the endoderm. Diploblastic organisms or organisms which develop from such a blastula and include Cnidaria and Ctenophora, formerly grouped together in phylum Coelentrata, but later understanding of their differences resulted in them being placed in separate phyla. So here they're saying blastula, need not get confused. The process of germ layer differentiation is seen during the gastrulation of blastula. So that is the reason why we use the word blastula, but need not get confused. It is very clear that during the gastrulation only the germ layers are being formed. So here is Hydra belonging to phylum Coelenterata. So if you take a transverse section, you can very clearly make out two layer of cells, an ectoderm and an inner endoderm. So therefore they are having only two germ layers and is a clearly a diploblast with two layer of germ cells, one outer ectoderm and an inner endoderm. If we take a vertical section of Hydra, there also you can make out the inner endoderm, the outer ectoderm, and whatever is written as Mg here is nothing but the mesoglia. This mesoglia is not a cellular layer, it's a non-cellular layer. So therefore, clearly there are only two layers, ectoderm and endoderm, making it a diploblast. The endoderm allows them to develop true tissue. This includes tissue associated with the gut and associated glands. The ectoderm, on the other hand, gives rise to the epidermis, the nervous tissue, and if present, also the nephridia. Simpler animals such as sea sponges have one germ layer and they lack true tissue organization. And group of diploblastic animals which are alive today, they include jellyfish, coral, sea anemones, and comb jellies. In this image, I have tried to differentiate the three layers: endoderm, mesoderm, and ectoderm, and the organ which or the tissues which is derived from it. That is, the endoderm gives rise to lung cells, thyroid cells, and digestive cells. And mesoderm gives rise to mostly muscles like cardiac muscle, skeletal muscle, smooth muscle, and along with it, it is also responsible for the formation of RBC cells and tubule cells. And ectoderm gives rise to skin cells, neurons, and also the pigment cells. So this is just an example. There are other cells which are derived from these layers, but these are the major ones. All the more complex animals from flatworms to human beings are triploblastic with three germ layers. A mesoderm as well as ectoderm and endoderm is present, therefore three layers, and then therefore they are called as triploblast. And mesoderm allows them to develop true organs. The triploblastic condition. In triploblastic condition, the blastula gives rise to three primary germ layers, that is the ectoderm, mesoderm, and the endoderm. Also, a fourth layer consisting of germ cells are set aside in the embryo at the blastula stage, which are incorporated into the gonads during organogenesis. The germ layers form during gastrulation of the blastula. Additionally, the term may refer to any ovum in which the blastoderm splits into three layers. Here in this image, you can see the three layers very clearly in the gastrula stage. You can see an outer ectoderm, an inner endoderm, and the middle mesoderm. Since it is having three layers, we are calling the condition as triploblastic condition. Also, a fourth layer will arise, which we are not calling it as germ layer, but rather it is contributing to the formation of reproductive system, to the formation of gonad formation. So all these germ layers are formed during gastrulation, which is very important. You should not get confused. Though we use the word blastula, it is the blastula which is further getting differentiated into gastrula, and the process is known as gastrulation. So during this gastrulation only the germ layers are being formed. All higher and intermediate animals from flatworms to human beings are triploblastic and they belong to Bilateria. Simple animals which are qualified as diploblastic such as cnidarians, including jellyfish, corals, and hydra, they possess two germ layers. Even simpler animals such as sponges contain no true tissues. The body wall of sponges consists mainly of two layers, the pinacoderm and the choanoderm, and a non-cellular mesohyle between these two layers. And triploblasts emerge from diploblasts, so they emerge within the diploblast. So it was the first diploblastic condition which evolved first, and further another layer of a cellular layer got added, leading to the evolution of triploblasts. In this image, I've tried to show the triploblastic condition under three different categories. That is acoelomata, pseudocoelomata, and coelomata. Let us learn about body coelom in the next session. But keeping it aside, understand coelom as the body cavity. And in case of a flatworm, say for example, planaria, you have three layers: ectoderm, endoderm, and the mesoderm. Similarly, in roundworms, like the phylum Aschelminthes, again three layers are present: ectoderm, endoderm, and mesoderm. And in phylum Annelida, example include earthworm. There also we have three layers: ectoderm, endoderm, mesoderm, making it essentially triploblastic. That is, any phylum, any an organism belonging to phylum Platyhelminth onwards are essentially triploblastic in nature. But the difference lies in the presence or absence of true coelom. In case of flatworm, it is having no coelom. Therefore, it is an example for acoelomate triploblasty. Whereas in case of roundworms, coelom is present, but a false coelom is present. Therefore, it is an example for pseudocoelom. So, roundworm is also an example for pseudocoelom triploblastic condition. Whereas from phylum Annelida onwards, true coelomic cavity can be seen. Therefore, it is an example for coelomate triploblastic condition or coelomic triploblastic condition. Here is a picture showing the increasing complexity of the three main body plans, that is the diploblastic acoelomate. All diploblastic animals, that is cnidarians and coelentrates, they are essentially acoelomic in nature. Therefore, we call it as acoelomic diploblastic condition. Whereas the next level is, the next body plan is triploblastic acoelomate. The example include Platyhelminth. And the next major body plan is triploblastic coelomate. And all animals from phylum Annelida onwards fall under this category. Whereas Aschelminthes is a triploblast essentially, but a pseudocoelomate, which is not a very major body plan, so it can be kept aside. All the phyla fit into one or the other body plan given in this picture. So we have the increasing complexity going in this direction. That is, the first one is diploblastic acoelomate, further triploblastic acoelomate, and then triploblastic coelomate. In diploblastic acoelomate, we have only two layers, that is endoderm and ectoderm. In the triploblastic condition, another extra layer called mesoderm has been added. And here, since there is no coelom, it is considered to be an acoelomate. But whereas further evolving complexity is the presence of true coelom, apart from having three germ layers, that is endoderm, mesoderm, and ectoderm. So the complexity increases from lower organisms to higher organisms. So to quickly summarize, a germ layer is a primary layer of cells that form during embryogenesis. The more tissue layers and the presence of a coelom, the more complex the animal is. Humans, for example, have a coelom and are triploblastic, making them one of the most complex organisms in terms of their body plan. And we have diploblastic condition, that is any animal possessing two major tissue layers, that is ectoderm and endoderm, are called as diploblasts. And triploblastic condition is where animal possesses three major tissue layers, that is ectoderm, endoderm, and a middle mesoderm. And such animals are known as triploblasts. Let us now quickly solve some multiple choice questions. The first question is, which of the following is an example for diploblastic condition? So diploblastic condition means essentially it has two germ layers, outer ectoderm and inner endoderm. So the answer is cnidarians. So the cnidarians are an example for diploblastic condition. Apart from cnidarians, the phylum Coelentrata is also an example for diploblastic condition. Whereas roundworms, annelids, and arthropods, they're all essentially triploblastic because they have an additional cellular layer called as mesoderm. Now the second question. During embryogenesis, germ layers are formed during dash stage: morula stage, blastula stage, gastrula stage, and neurula stage. The answer is gastrula stage, because during the process of gastrulation, where the blastula is converted into gastrula, you can find the germ layer. So essentially the answer is gastrula and not blastula. Third question is, identify the triploblastic acoelomate. So they're asking triploblastic condition. So all these examples, okay, we have to analyze. But they're also saying acoelomate, so the one which lacks coelom. So here is the catch. Not just identifying the triploblastic condition, but also identifying the acoelomate. Coelentrate, first option. Coelenterata is a diploblast, so clearly Coelenterata can't be the answer. The second option, Platyhelminthes. Yes, it is triploblastic. Also, it is an acoelomate because it does not have any coelom. So, therefore, Platyhelminth is the answer. Whereas the other two options, Aschelminthus and Annelida, Aschelminthus is a triploblast, but it's a pseudocoelomate, not acoelomate. Whereas Annelida is a triploblast and a true coelomate. So, even Annelida can't be an answer. So, the answer is Platyhelminthus is a triploblastic acoelomate. And the fourth question is, epidermis is derived from which layer? Ectoderm, mesoderm, endoderm and periderm. So we know that among the germ layers, it is the ectoderm which gives rise to skin. That is the epidermis. So answer is ectoderm. And the last question, choose the odd one out. Coelenterata, Platyhelminthus, Aschelminthus and Annelida. So here the question is asked with respect to the germ layers or the condition. Of all these four options, 3 are triploblastic and 1 is diploblastic. So essentially the answer is Coelenterata. So Coelenterata is a diploblast, whereas the others are triploblastic in nature. So therefore it is an odd one out. I hope this session was useful and thank you for your patient listening. Have a great day.